So to summarize everything that we talked in the previous part of the lecture, remember, when you start with a heterozygous during meiosis and you have this combination, depending on the distance, how close or how far are the genes, there are different frequencies of recombination. So, so in the following slide, I'm going to show you again what do you think that happened with the genes A and B. Yeah, so what do you think that happened here? And what do you think that happened between A and C? That they are much far from each other, and what this means. So I might be a little bit repetitive, but I want that you remember this. A and B, they will have no recombination, and I will show you in the following slides multiple times the same thing. Why? Because they are too close to each other, so there will be no recombination, so they will be linked, they will inherit together. If two genes are too close, it's too difficult to break them because they are just too close, and the process of recombination, we will not be able to mix it. On the other hand, when you have the genes are relatively different distant, like A and C, you can go to different amounts of different frequencies of recombination. Yeah, so for sure it will be higher than zero. Yeah, because zero it will be when there is A and B, when there is no recombination, and it will be lower than 50%. Why? Because 50% will be what we get when they are have independent assortment, when they are in different chromosomes. Okay, so in the following slides, I'm going to show you these ideas about how, depending on how close the genes are, they will find different degrees of or different frequencies of recombination. So now we're going to talk more about this, and I'm going to compare link versus non-link genes. And I'm going to show you this diagram again to try to make things simpler. So here you have a link genes that I want to that you observe the distance. Here you can see two examples of a non-link genes. So here, first one is very easy. The first example, as we talked last in the previous slide, link genes sit close to together on a chromosome. As a result, make them likely to be inherited together. So this is very straightforward. Now, in the second case, it's also very straightforward. Genes that are separated in separate genes on separate chromosomes are never linked. This example is very clear. However, the third example, so let me put here second, and here I'm going to put a third. Here in the third case, you are in between 1 and 2 because the genes can be they are a distance, and they can be linked or not linked. So they can be far from each other, that they can be not linked, or they can start getting closer and start getting linked. So remember in the third example, but not all genes on the chromosome are linked. If they are far enough, they are not. They can have they can be crossover, so they are not be linked together. So remember, genes that are farther away from each other are more likely to be separated during the process of called homologous recombination. And what we are going to do in the lab is try to study some examples of homologous recombination and see how you can create different combinations when you have chromosomes that are, well, sorry, when you have genes that are on the same chromosome. So remember again this example here, when you have genes with recombination frequency then less than 50%, they are on the same chromosome and they are considered linked. However, in addition to the gene 1 and gene 2, you can have genes that are at a different distance on a different chromosome, and this is why it changed the recombination frequency, but they are always going to be less than 50%. And all these blue genes, And these ones, they are called linkage group, that are all the genes that are on the same chromosome. Here, as we said, they are the genes that are not definitely not never going to be linked because they are on different chromosomes. And in these cases, two genes that undergo will undergo under independent assortment, so have a recombination frequency of 50%. And they are located in non-homologous chromosomes or far apart on the same chromosomes, so they will be always unlinked. So how this process will take place? So here again, non-link and linked gene, and there is a process of meiosis. And if you see, you have A genes in yellow in this chromosome, and you have this in this chromosome that is orange. They will be the chromosome will be in orange, and in both process, in both in the link and non-linked, the same process is happening. Here there is meiosis in both, and there is a mix between the yellow and the orange. However, as in the non-link, as you can see here, you have here the parentals that are the same, but you have neallele combinations when A and B are together, because there was the process of recombination. So you will find orange and also yellow. On the other hand, in the linked genes, the same thing process is happening. You have A and B, and here you will find a combination between both of them, but between orange and yellow. But here there will be no new allele combination. All of them will look the same. All the V will be here, all the A will be here, and same thing happening in the other ones. So to summarize, uh, I show you that when you have a gene 1 and 2 that are far apart, it's likely that a recombination event will happen. So as I show all this in this example. On the other hand, in the genes 3 and 4 that we saw in the linked genes, uh, as the genes are sitting so close to each other, the recombination event, it will happen. If it happens, it will be less unlikely. So it will be closer to zero. So the closer they are, the closer to zero that they will be, those processes to take place. So in other words, and we will do that in the lab, when we make the crosses, we will find that when the genes are very close to each other, if we take 1,000 flies, we will find one or two flies that have in genes that are very close, so it's very close to zero, and in genes that are relatively far from each other, instead of finding one or two flies, you will find 20 or 30. And in that way, we is how we start mapping the chromosomes, we start finding the distance between chromosomes, even without need to sequence, because at the time there was no way to sequence the chromosomes. Okay, this is the last example, the non-linked genes when they are in different chromosomes. So if you see, these genes are on separate chromosomes, so they are never linked. So here, although there is still recombination here, and you have orange and yellow chromosomes, this will not have any effect. So here you will have all the A's, all the 5, and here you will have all the 6, but this process will be determined at random. And to summarize this, because there is nothing holding them together, the alleles can pass to gametes in any combination, and this is a representation of all the potential combinations between 5 and 6. And to summarize all these ideas, I just put all this comparison between link and unlinked gene, that you can, if you want, you can take notes, and maybe it could be helpful for you. This is the second part of that table. Again, if you want, you can take notes or not.